Okay, so we have this short 10-slide presentation on mutations. Most of it's review. You should remember it from our basic material. If you really paid attention in week 2, you set yourself up well for the rest of this course. If not, go back to that material. There's a lot of it, but it does provide a foundation. All right, so our learning objectives for this very, very tiny presentation are to review common types of mutations and understand their impact on protein synthesis and protein function. Don't get nervous. We're not going into deep detail. But then we're going to discuss how mutations occur and how they can be repaired. We'll identify common mutagens. Those are those environmental agents or toxins that cause alterations in the genome and therefore human health. And we'll discuss how mutations don't necessarily lead to health problems. Not all of them are harmful, and they are common. And then we'll apply some earlier content to understand how illness develops from things like deletions, inversions, and duplications of chromosomal material. Usually it's a partial chromosome that's involved, but there's multiple genes maybe on that piece. So let's review some basic concepts. You know, a mutation is a change to a genetic material, to a piece of genetic material. Could be a single base, could be a whole chromosome. Mutation is a change. Somatic mutations happen in your diploid cells. They are limited to individual cells or tissues. They are not inherited by offspring. If your body's good at repairing cells with changes to their DNA, they don't cause problems. Germline mutations start in the sex cells, and that can happen during meiosis 1 or 2, and they are found in every cell of an offspring. They are passed on through generations. They become heritable mutations. And we have some other terms, point mutations. Point mutations are like a single base, a single nucleotide pair. You substitute a T for, a thymine for an adenine, and now you have a different base pair. These are the ones we talked about in week 2. And we have 3 basic types. If you want to go back to that material, you can, but this is what you need to know. There's silent, no change on protein formation, because either the gene does not code for a protein, it's a non-coding gene, or the protein can still be made, even though there is an error in that code. It will be corrected in the formation of the protein. Missense. There is a protein that comes out of it, but because the code has been altered by a single base pair, we now have a different protein, a different amino acid, or maybe the same amino acid in a different order, codes for a different protein. And now we have a change in the structure of some enzyme or cell wall or something. That's a missense. Nonsense means the protein can't be completed because there's a stop codon. Something that doesn't make sense stops you from making that protein. Missense and nonsense mutations can lead to health problems, but sometimes not. So, point mutation, single base is substituted. I know it's a little repetitive, but doesn't hurt. The bases are still in triplet form. Remember, they get read in like triplets. There's like 3 bases, and the RNA picks it up. So it has to be in multiples of 3 to make a protein. So an example of a single base substitution point mutation, sickle cell disease. So a single base substitution results in a missense mutation. A protein is formed, but it's a different protein. We have valine, the amino acid valine, instead of glutamate. So we have a red blood cell, or we have that beta globin protein, but it folds. And now it carries less oxygen, and it gives that cell a sickle shape to it, where it gets stuck in the joints, and we have sickle cell disease. That happens from a single base substitution that leads to the development of a whole different protein that doesn't have the structure of a normal beta globin protein. That's a point mutation. In contrast, we have frameshift mutations, where the DNA is disrupted in such a way that bases are added or deleted in numbers that are not multiples of 3. And you get this crazy pattern that RNA can't read. So no protein is synthesized by the gene that's supposed to do that. For an example, we have BRCA1 gene. Normal gene codes for protein that protects that person from breast and ovarian cancer. But if you inherit an abnormal allele with this frameshift mutation, that protein is never produced and the person is more susceptible to breast and ovarian cancer. Because cancer is a complex disease with many mechanisms that influence its development, it's not a guarantee that if you have that allele, you will get cancer. But it does increase the likelihood. One less protective mechanism is in place because that protein isn't formed. All right. So that's the difference between like a frameshift mutation and a point mutation. Mutations can occur on any genetic level. It could be at the individual nucleotide, one base. It could be a whole segment of DNA. It could be a gene. It could be a piece of RNA that reads the DNA and then codes for a protein. It could be a part or a whole chromosome. It could be the whole genome. So a mutation is just a change to DNA. It can affect small amount or large amount, and it can occur in any process. It can occur most especially when DNA is being copied. Sometimes it is copied in error. You have extra material or something's missing. There's deletions or insertions or whatever. Or it can happen during division where the actual material kind of breaks off or is duplicated when those cells are, when that cell finally divides and repackages its duplicated material into two new cells. It can happen during protein synthesis. So mutations can come spontaneously, just as an error in the process, or they can be induced by exposure to environmental toxins called mutagens. Mutagens that cause cancer are called carcinogens. Mutagens that cause embryonic disruption are called teratogens. Probably a little familiar with both of those terms, but I'm just repeating them. The location of mutations is not entirely random. There seem to be hot spots or weak points where the DNA is a little more fragile, a little more susceptible to being changed. Mutations are more common when telomeres are shorter because those end caps don't protect as much material. But there do seem to be breaking points where changes are more likely to occur. And repair mechanisms are less likely to identify and correct the mistakes. So, how does DNA repair itself? Well, DNA has to be a high fidelity process. For life on Earth to continue, our DNA has to be able to copy itself and reproduce and do so with a high degree of faithfulness to the original model. So, there are a lot of built in editors in DNA, a lot of genes that regulate, code for enzymes that help repair protein. So these are like little mechanisms that identify and correct errors, similar to your Cas9 protein that we talked about with CRISPR. So you have enzymes and they read the DNA. When they find an error, they clip it out and replace that incorrect information with the correct bases. And that is a really, really efficient process. Only about 1 in 1 million errors slips through. It can vary from individual because DNA repair, the efficiency of your DNA repair is an inherited trait. Most people have average. They manage their day-to-day random errors as they age. That process gets a little sloppier. But they function for a number of years. They live and they kind of continue through life. The little errors that slip through get repaired until they start the aging process, and then you see more mutations. Some people have greater than average function in their DNA repair mechanisms. They have these like longevity genes. If you want to get really dorky, you can read a book called Lifespan. And that talks a lot about the different repair mechanisms in DNA. So these people have longevity because their repair mechanisms are like super police. Um, and they repair that DNA and keep it from making changes. Some people have very poor repair mechanisms and they have additional errors. Um, so these folks are more prone to diseases of, whatever type. So if you have poor repair mechanisms, you're more susceptible to a number of diseases at younger ages. However, it doesn't matter how efficient your inherited DNA repair is if you overwhelm it with exposure to environmental mutagens like tobacco smoke or sunlight. You will get direct damage to the DNA and it will overwhelm those repair mechanisms and you'll have health problems. So that's that. Common mutagens. This is probably not a shock to many of you. X-rays are both teratogenic and carcinogenic. So ionizing radiation disrupts DNA. Sun exposure, especially to cells that are exposed, like skin, and the cells of the eyes can be damaged. It will lead to increased rates of diseases associated with aging over time. We have chemical agents such as PAHs, polycyclic aromatic hydrocarbons, and they come from like over grilled meat. We all like that nice crust on a steak if you eat meat. Um, those are found to be carcinogenic. Mustard gas, so it was used as a chemical agent in warfare and then in smaller doses was used as an alkylating agent for cancer treatment. That can cause changes to the DNA. Bisphenol, phenolic acid, BPA. I might be saying the P more wrong and kind of drawing a blank on that. But BPA is found in a lot of plastics, and it does have mutagenic effects. Nitrates in your processed meats, bacon, hot dogs, people love them, but they do in amounts that are, enough to cause changes in the DNA. They can cause cancer. They are carcinogenic. Pesticides, organophosphates, and DDT. We have talked about those before too. I think when we talked about social and environmental justice with malaria. So those things disrupt DNA, cause mutations at different levels. And we have biological agents like viruses and bacteria, and the resulting inflammation causes changes to the DNA. Now, it's not all bad news. There are some things that protect you from mutagens. Antioxidants, which are plentiful in fruits and vegetables. We also make one of our own antioxidants called glutathione. Not really essential content for us, but if you're interested in preventing disease, it's worth a try. You can look at that, ways to promote glutathione development. But really, if you get antioxidants into people, plant-based foods are full of them. Some animal-based foods have them, but more commonly, we're looking at vitamin A, vitamin C, vitamin E, flavonoids. So your nuts, your fruits, your vegetables, seeds. A lot of herbs have anti-mutagenic properties. The more you can encourage that healthy plant-based, people can eat animal foods, but not to the exclusion of all these good fruits and vegetables they should be eating. Non-harmful mutations. Okay, so like I said, we're kind of moving right along. Many mutations have no effect on the individual. So the impact is minimal if the region of DNA is non-coding region. If it doesn't make a protein, it could be changed. It's really just not going to matter. We're all exposed to mutagens all the time. If we develop somatic mutations in a limited number of cells or tissues, there's usually no impact on functioning. Those cells will be programmed to commit suicide. There's all these pathways that will induce apoptosis. Might see that again. We didn't test on it for the first exam, but you might see it in the second. So apoptosis is that cell death. It kind of commits suicide to save everybody else. Very noble act, if those cells are abnormal enough. And occasionally, we see mutations that have benefits to the person who has them. For example, there is a mutation that prevents white blood cells from getting infected by HIV. So individuals who are exposed to HIV, if they have this mutation, they will not contract that disease. That's kind of how, it's theorized that evolution occurs. That eventually some mutations are beneficial enough to give people an advantage in reproduction, and that gene will be passed on, that changed gene. All right, we are going to finish up with deletions, duplications, and inversions. These are bigger scale mutations. They involve larger segments of a chromosome and multiple genes. They're usually described in terms of a location like 13q14 or 1q11, 1q22 deletion. So you'll get the number of the chromosome, the arm of the chromosome, and then there's like a locus, like a little location on that chromosome. And then it'll tell you whether it is a deletion, a micro deletion means small amount, a duplication. The description of that mutation will include all that information. That's why it was like I included information about P arms and Q arms, just so you know what they're talking about. It's like a map to the place where that mutation occurred. And like I said, there are like hot spots or breaking points where it's more commonly occurring. So in this category, we have deletions. And that's where we're missing genetic material and we have reduced expression of a trait. So maybe on the long arm of a chromosome, we have a fragile spot, 13q14. It breaks off, does not get repaired, and we end up with a child who has a retinoblastoma. So there is a gene that controls tumor suppression on chromosome 13. It is specific to tumors that happen in the retina of the eye. If you have a deletion, you only have a single gene. You get like a half a dose of that protein synthesis power. So as a result of that deletion, children can develop cancer of the retina or a retinoblastoma. That's what a deletion does. It reduces the expression of a trait, and most traits are going to be to code for a protein. So there will be less protein synthesized in a deletion. If we have a duplication, that is extra genetic material. So sometimes that happens. Try to visualize this, where I couldn't find a picture of it. The DNA kind of folds and sticks on itself. So the RNA reads it and then kind of repeats it. So we have things like Charcot-Marie tooth syndrome, where you get more of a dose of the gene that codes for this protein called PMP22. Now, that protein in excess will cause a breakdown of the myelin sheath around the nerves. So in Charcot-Marie tooth syndrome, the result is that you have weakened nerves and decreased motor function. If you have the right dose of that gene, if you have just the two alleles with the normal amount of bases, they will code for a normal amount of protein, and it just kind of makes sure that everything's functioning. If you have extra protein, it breaks down the myelin sheath. And that's the result. Okay, inversions. You have a piece of a chromosome that breaks off. Remember when we talked about translocations and you had a piece that broke off at a weak spot and it attached to a non-homologous chromosome? Well, an inversion happens when it attaches back to its original chromosome, except that now it's upside down. And that usually happens as a de novo mutation. It's not usually inherited, but it happens in the sex cell and then it becomes a germline mutation. So DNA can be lost or it can be added in the process. Sometimes it's all there, it's just upside down. One example of an inversion is chromosome 17q12. So it's on the 17th chromosome, on the Q arm at the locus 12. It ends up in a microdeletion. So a piece of chromosome 17 breaks off and reattaches itself upside down, but a little piece of that material is lost, and we have renal cysts and diabetes, RCAD syndrome. There's other features that go with that because there were other genes on that little strand of the chromosome, but the defining feature that most people have is the renal cysts and diabetes, and it's called type 5 diabetes. It's linked to a higher incidence of autism, schizophrenia, and intellectual disability. but some people can have that mutation and have no symptoms at all. So that's an example of an inversion. And let's see, I don't think there's any more to this slideshow. That was just like a basic primer. As we move forward, we're going to talk about conditions that we might see, such as autism or schizophrenia or other mental health problems, cancers. So these terms will be important to you. It's just good to have like a really basic understanding of how genes change and how that impacts health. All right, have a good night.